I'm not so fine because today is a sad day. Our one of the very famous rice scientist, Dr. Tomal Lota Aditya, suddenly died. She was 1 year senior to me. She was the director of research, Bangladesh Rice Research Institute. So we express our uh, uh, deep, deepest sense of sorrow on her death, and she was massively attacked by in her heart. And yesterday we had a whole day biotechnology review workshop at DRC with her, and I had lots of memories. She asked me. I always called her Dhan Konna because she developed a large number of rice varieties in Brie, uh, which helped us to be self-sufficient in rice production in Bangladesh. So Bangladesh lost a great scientist last night. So mentally, I am a little bit depressed. Yesterday, when I called her during my speech, Dhan Konna, uh, Dr. Tomarlota Aditya, after the my lecture, she asked me to give a certificate from the IBG that she is a Dhan Konna. Uh, she was a very simple lady and received PhD from Imperial College of London. In her student life to the research career, she was outstanding. And she was a biotechnologist, but posted in the Department of Genetics and Breeding Department, Division of Breeding of Breed, and she led breeding department very successfully and received Bangabandhu uh, award on success, outstanding success in agriculture. So we express our deepest sense of condolence to her family. So anyway, I would like to start your talk today on DNA repair mechanism. Meanwhile, you know what is DNA, what is RNA, what is gene, what is genome, what is proteome, what is metabolome, what is transcriptome, and how amino acid is coded by genetic code, and different kinds of proteins are synthesized in the cell to do the function, and what is gene expression. So basic uh, uh, basic principles of biotechnology and genetic engineering, you received a lot of information. Uh, Now the question is whether DNA in our cell uh, is, prone to damage or not. Because if DNA is damaged, then we cannot inherit, and human Homo sapiens cannot continue as Homo sapiens. There was a belief that DNA never be damaged, or DNA never be changed in the early age. But later it has been found that DNA has DNA is prone to uh, cause damage by different environmental factors. Now the question is, if DNA is changed due to environmental stresses and other reasons, then how can we maintain Homo sapiens, or every species maintain their species traits? DNA has some, a cell has some inherent mechanism to repair and maintenance of DNA. When a new DNA is, uh, prepared in the cell, that is when DNA is replicated in the cell, uh, there are a system for proofreading whether the new DNA, daughter DNA is uh, exactly same to the parent DNA. So proofreading mechanism, DNA repair mechanism. If somehow one nucleotide or several nucleotide, one base is damaged, by any environmental factors like UV radiation or other radiation, or you are exposed to the carcinogenic substances.
স্যার
Yes.
স্যার কথা বোঝা যাচ্ছে না, একটু জোরে বললে মেবি ভালো হতো।
How about others?
স্যার
Others?
সবাই কাজ করছিলাম।
স্যার কথা একটু স্যার কমই শোনা যায়, আর স্যার একটু আসতে গেলে ভালো হয় স্যার।
হ্যাঁ স্যার,
মানে
সাউন্ডটা
একটু
অনেক
স্লো স্যার,
ধীরে আসতেছে স্যার।
একটু স্লো গেলেও ভালো হয় স্যার।
All right. I was talking about the DNA repair mechanism in the cell. DNA, when replicate in the cell, it makes its own copy exactly like the parent. But if DNA is exposed to our cell, or we are exposed to different UV radiation or some other radiation or some other carcinogenic substances, DNA may be damaged. In that case, it may be damaged at the base level, single base, or it may be damaged at the nucleotide level. Several nucleotides may be damaged. In that case, DNA, if DNA damage is continued and new replicated DNA contains the damage, then it would be detrimental for the organism as well as the cell. In that case, DNA repair and maintenance is necessary. In the cell, inherently, there are mechanisms for detection of damage occurs in the DNA, newly formed synthesized DNA, and then repair it by using some DNA repair mechanism. So it can be at base level, single base repair, or multiple nucleotides repair. And in the old days, scientists thought that DNA are not damaged by any other external factors. That means DNA is stable molecule, and they thought reasonably that this is why every species are maintaining their continuing as a unique species. But practically, DNA is prone to cause damage by different external, even in the internal factors. And scientists discovered that DNA has, a cell has the mechanism of repairing and maintenance of the DNA. And you may ask question, the repair and maintenance, is it 100% efficient? No, not 100% efficient. Sometimes the efficiency slightly poor. In that case, if a damage remain as damage, for example, one single base damage is continued during replication of new DNA, that means it is a mutation in the DNA, a single base mutation. It may be harmful or maybe not harmful. Single base mutations sometimes result serious consequences, even causes cancer, or sometimes no effect. Whole life it may be continued. Over the years of evolutionary process, different organisms, you can see in different organisms, in different species, among the species there are huge diversity. These diversities is due to the mutation resulted by the failure of DNA repair and maintenance by the inherent system. And for example, in human being, we have now 7.3 billions of population on this earth. And among the 7.3 billions of population, we are Bangladeshi. We have some unique characteristics. There are some Africans, there are some Mongolians, there are some Europeans, every nations, they have some characteristics, and these characteristics is associated with the mutations occurred and stably maintained in the DNA replication process. So today I shall elaborate what is DNA repair mechanism, what are the mechanisms, and why DNA damages are taken place. Let me go to the slide. So today's talk is DNA replication and repair, because DNA must be replicated before the cell division. Otherwise, cell would not get same DNA as its parent. DNA, a mystery of life, already you know that. From one cell to another, from one generation to the next, it continues as intact. The genetic information that governs how human beings are shaped has flowed through our bodies for hundreds of thousands of years through the DNA. It is constantly subjected to assaults from the environment. That means DNA is subjected to assault. Assault means under stress from the environment, yet it remains surprisingly intact. Replication, DNA replication is very crucial and essential step for the growth and development of any organism as well as reproduction. So in replication, there is a starting point, there is a finishing point. Must be ready before DNA replication. DNA must be ready, must know where to start. And in finishing, must ensures that each piece of DNA is replicated only once, therefore must know where to finish. So it is a very long double helical structure. So starting and ending must be very correct. And replication of DNA, actually when Watson and Crick first discovered the double helical structure of DNA, he actually understood that DNA must replicate in the cell before cell division. And the accuracy or fidelity is very important. Proofreading or repair must be distinguished between original and copy. That means proofreading, when DNA is one DNA is replicated to another, there is a mechanism of proofreading. Check one by one all the nucleotides, whether they are exactly same to their parent or not. And the accuracy is very important. And in this paper I shared with you, it is mentioned that DNA replication must taken place, and it was noticed by Watson and Crick. So DNA, cell cycle, cycle. That means in our body, cell has a certain age. For example, you have 90 years of total lifespan is 90 years. During this period, every cell, has a certain cycle. For example, our blood cell, after 3 months or 4 months, they are changed. In that case, new cells are needed to be synthesized or during your growth. So cell cycle has several phases. For example, S phase. S phase, starting phase, DNA synthesis is taken place, and then growth phase, that means G2 phase, and then mitosis taken place, and then G0 phase, and G1 phase, growth phase 1, and then cell completes its growth, and again the same cell can be replicated. So cell has several steps to follow for making a daughter cell which exactly carry the same genetic material. So before a cell starts to go for production of the daughter cell, it must synthesize the DNA in the cell. So start must be ready. G1 must all start at the same time. G1 to that means all must be started in the same time, or not one by one. Must know where to start, origin of replication, and then finish. Must all finish. That means complete S. Must ensure that each piece of DNA is replicated only once, therefore must know where to finish. That means the replicon. This is very, very crucial in the cell, and cell has such a memory to do that. So accuracy is very important. Sometimes not 100% accurate. In that case, problem generates. Must distinguish between original and copy, which is called epigenetics. That means there is a proofreading to check the newly synthesized DNA whether same to the parent or not. So DNA replication occurs during S phase of the cell cycle, semi-conservative in nature, and it starts from 5' to 3' directions in the DNA cycle, so starting point is 5' to 3' in the sugar unit, and leading strand, lagging strand, RNA primer, these are very, very important. So single origin in bacteria, multiple origins in eukaryotes, because bacteria is unicellular, but in eukaryotes like human, multicellular origin of start is multiple, bidirectional and autonomously replicating sequence elements from yeast cloned origin. Scientists already studied a lot on yeast, the model eukaryotic organisms in molecular biology. So how it taken place? DNA replication. During DNA replication, DNA is a double helical structure. This is the parent duplex, DNA. But before replication, it uncoiled and become open a double helical structures, and then complementary sequence fill up the single strand and make the double strand. As a result, from one DNA, two daughter new DNAs are synthesized. And it starts, I told you, from how it starts. Here you can see 3'. It starts to fill up the gap, and DNA synthesis always occurs by adding nucleotides to the 3' OH of the growing strand, and continuously fill up the nucleotides. And synthesis is always in the 5' to 3', 5' to 3'. This is the principle scientists already discovered in case of DNA replication. Here, a very simple way. This is a double helical, DNA structure. Double stranded DNA structure, not helical. It is non-helical for giving you a clear idea. And it is unzipped, this part and this part, and then this single strand from 5' to 3', it goes to fill up by nucleotides synthesized in the cell. And then from 1 to 2 DNA is synthesized in the cell. Here more, clearly it is shown how one by one, nucleotides are added, and, several enzymes like, primase, gyrase, they are involved in DNA synthesis. So this is the replication fork, how it is taken place. So replication enzyme, for replication, synthesis of any biomolecules in the cell. Enzymes, I told you, enzymes are protein, but all proteins are not enzymes. Enzymes have the catalytic principle. Enzymes are involved in synthesis of molecule in the cell. So what enzymes are involved in DNA replication? DNA polymerase matches the correct nucleotides, then joins adjacent nucleotide to each other. So DNA polymerase detect the correct nucleotide, which is complement to the single strand, and place it in the right place and make the double-stranded one. So polymerase means make polymer, bigger molecule. Primase provides an RNA primer to start the polymerization. So it is also crucial to start the DNA polymerization. Another one is ligase. Whenever you heard about this enzyme, ligase means sealing something. It is pasting something, joins adjacent DNA strand together, fixes or nicks. So ligase enzyme is very important in genetic engineering. We also use this enzyme. So helicase enzyme, another enzyme, unwinds the DNA and melts it. Before replication, unwinding is necessary, and melt is necessary. So helicase is involved in making the single strand binding proteins. So there are some proteins involved in single strand binding. Keep the DNA single stranded after it has been melted by helicase. So helicase melt and unwind the double strand, and then helicase involved in single strand after it has been melted by helicase. That is, proteins are some single strand binding proteins are involved, and to keep it open, single stranded, like I showed you, unzipped, uncoiled part, and so that new nucleotide can be added. Gyrase, an enzyme within the class of topoisomerase, and is a subclass type of type 2 topoisomerases that reduces topological strain in an ATP dependent manner, while double-stranded DNA is being unwound by elongating RNA polymerase. So during elongation of the DNA through the polymerization, this gyrase enzyme is involved, and it maintains the unwound. That means no wounding in the DNA. Correctly, polymerization taken place. So helicase, gyrase, these are the enzyme, and telomerase finishes of the ends of DNA strand. So this enzyme, at certain point, it whistle that DNA polymerization is finished. And this is the, how several guards, several involvement of enzymes, they do the correct replication of the DNA in the cell. And it is very essential. This is why we can maintain our DNA as human DNA. Day after day, our DNA is damaged by UV radiation. This is a big question. That means DNA is damaged by UV radiation. Free radicals. Free radicals, have you heard about it? In our cell, during the stressful environment, when you are under stressful situation, for example, when you have fever or infected by any pathogens, for example, SARS-CoV-2, a cell produces lots of free radicals. What are free radicals? For example, hydrogen peroxide, superoxide, or singlet oxygen, or some other free radicals which are very toxic, and they are used as defense radicals against the the pathogen. But these free radicals, when excess is produced, then they can damage even our own DNA. So in severe stressful situation, either biotic and abiotic stress, free radicals are or reactive oxygen species or superoxides are produced in our cell naturally. But there is a mechanism of minimizing, detoxifying these free radicals. It is called, antioxidants. In the body, there are some enzymes that can, detoxify these free radicals. But even though when too much free radicals, then detoxification mechanism may fail. In that case, DNA may be damaged. For example, if you are exposed to severe radiation, radioactive substances like UV radiation, in that case, huge free radicals are generated and your DNA may be damaged. And some carcinogens, for example, chemical substances, if you are exposed to some chemical substances, even, genobiotics, that is human-made chemicals, even some antibiotics, they can be carcinogenic. Carcinogenic means they can damage your DNA. And why the term carcinogenic? Because it is linked to the cancer. Stable DNA damage may result cancer in the human and other animals. But despite all that, our DNA at molecular level is inherent. That means it can maintain its own structure. So DNA have the maintenance and repair mechanism, obviously. Otherwise, how can it does? Thousands of changes happens in our genome at cellular level. But these changes affect our cells when process that happens thousands of times a day are divided. So when cell divisions are taken place, DNA replications are taken place, in that case, thousands of damages are taken place in the DNA, but thousands of repair and maintenance are also taken place. The reason why our genetic material is not destroyed by chemical levels is due to the large number of molecular systems that control and repair continuously our DNA. So it is now clear to you that in our cell or cells of other organisms, there are repair and maintenance mechanisms that continuously repair if any damage has taken place during a cell division, or even in the lifespan of the cell, and maintains the continuity. But 100% this repair maintenance is not correct. And this is why mutation taken place and diversity of organisms we can see in the earth. In 2015, a Nobel Prize in Chemistry was won by some scientists, Thomas Lindell, Paul Modris, and Aziz Shankar. He was basically, Turkish origin, a Muslim scientist. They got Nobel Prize for their discoveries on mechanistic studies of DNA repair. Mechanistic studies means how mechanically DNA repair is taken place in the cell. These three guys discovered the mechanism, not recently. They discovered, many years ago, but they got the Nobel Prize in 2015. Damage of the genetic material poses a threat to all organisms. To counteract this threat, cells have evolved a series of intricate DNA repair pathways that correct DNA lesions. Lesions means damage, affecting base repairing or structure of DNA. So base repairing means if single base is damaged or damage is limited to single base, then replace the wrong base and add a correct base to maintain the DNA structure. And it can be a bigger, a nucleotide level. So some surgery is done by the mechanism, repair and maintenance mechanism in the cell. So DNA repair and maintenance is really fascinating, and scientists discovered and got the Nobel Prize. Today, we understand the molecular mechanism of these pathways in great detail, in large part due to the pioneering studies by Lindahl, Modris, and Sankar that opened up the field of DNA repair mechanism. It is now a very big area of scientific research. They awarded for having mapped and explained how the cell repairs its DNA and safeguards the genetic information. So these are the guys, you can see their face, and if you visit the nobelprize.org, you can find them as the winners of chemistry prize in 2015. So in human genome, encodes the information needed to create a complete human being. You already know. Not only human genome, in case of our frog genome, encodes all the information needed to create a complete frog. So all organism has like this. During every cell division, more than 3 billion DNA base pairs are replicated, and copies of the genome are transferred to the daughter cell. That means human genome contains 3 billions, about 3 billions of bases, and all are necessary to correctly, replicate. And the DNA replication machinery responsible for this task still makes occasionally mistakes. Occasionally mistakes, I noticed several times in today's lecture that occasionally they make mistake. Given the size of the human genome, the large number of cells in the human body will inevitably accumulate during the lifetime of an individual. Most of these errors that means mistakes taken place during DNA replication will remain silent. Most of the errors, no symptom expressed, no harm to the human, but they can also cause serious diseases like cancer. So cancer is actually due to the mutation taken place in the DNA in any cancer. So it is one kind of genetic disorder disease, not non-communicable disease, not caused by the pathogen. Despite its essential role in storing genetic information, the DNA molecule has limited chemical stability, is subjected to spontaneous decay. Processes such as hydrolysis and oxidation occur at significant levels in vivo, in part due to reactive metabolites continuously generated in various physiological processes. I'm really sorry, in the upstairs, someone is doing this, making this noise. So in addition, external factors like radiation and genotoxic chemicals will further stimulate of DNA damage formation. That is, external factor like radiation and genotoxic chemicals that can affect the genetic material like DNA, RNA. Okay, the inherent instability of DNA constitutes both an opportunity and threat. What are the DNA lesion can block important cellular processes such as DNA replication, transcription, cause genome instability, and impair gene expression. Lesions can also be mutagenic, change the coding capacity of the genome, which can lead to devastating diseases and conditions associated with genome instability, including cancer, neurodegenerative disorders, or biological aging. Biological aging means you can, some people can quickly become, old due to the DNA damage. Furthermore, mutagenic chemicals, radiation can also have a healing effect. They can also, for instance, be used to treat cancer by introducing DNA lesion that halt cell proliferation and stimulate programmed cell death. This is very interesting and maybe contradictory for you. DNA damage by using chemicals, mutagenic chemicals, is harmful, generally harmful for us. But some chemicals, if you can use to treat the cancer, because cancer cell has, as the DNA, which is already damaged. If you can somehow target the DNA of cancer cell and can damage the DNA, then the replication of cancer cell you can stop and you can cure the cancer. So it has some advantages, that is mutagenic chemicals. The cell has developed ways to counteract DNA lesions and to keep DNA mutations as a tolerable level. A number of different DNA repair mechanisms correct lesions and safeguard the integrity of the genome. Already I mentioned this statement, and I am repeating so that you can understand what I am going to tell soon. 4 fundamental DNA repair pathways delineated by the Nobel Prize laureates will be discussed today. So if I ask you simply a question, a simple question, what are the causes of DNA damage? DNA can be damaged by chemical mutagens, radiation, and free radicals. Free radicals is endogenous, not external. That means mostly endogenous. So in your cell, some superoxide or reactive oxygen species, they are produced to defend any enemy if you are exposed to any stress. But if too much is produced, then body detoxification mechanism cannot minimize or neutralize them. They can also cause the damage in the DNA. So energy decomposition in DNA damage. For example, if you are exposed to radiation, DNA receive too much energy from the radioactive material, and DNA damage taken place. How it is taken place, here you can see hydrolysis can be taken place, and DNA, for example, damage is taken place due to absorption of energy and radiolysis of water. So as a result, you see DNA damage is taken place in these 2 parts. So radioactive materials can cause the DNA damage. So respiration and aging, carbohydrate and oxygen, they are burning in our respiratory process like TCA cycle or glycolysis cycle, and makes carbon dioxide and water and energy. But sometimes it can produce hydrogen peroxide or hydroxyl or superoxide molecules. They can these are free radicals. These they can cause damage and cause a lesion in the cell per day, and it is happening, but most of them are repaired. So despite 1,000 times of alterations that occur in DNA each day, that means in our DNA 1,000 times of alterations taken place, few are retained as mutations. Most of them are repaired. Efficient repair mechanisms exist in the cell. Importance of DNA repair highlighted by number of genes devoted to DNA repair. That means some genes are involved in expression and doing the job of DNA repair. Mutation rates with inactivation or loss of DNA repair gene. So defects in DNA repair associated with several disease states. Here is the DNA replication and repair disorders. You can see the frequencies. Disorder like Fanconi's anemia due to the deficient excision repair, base excision repair, if taken place, if deficiencies is taken place in base excision, then 22,000 of population, 1 person suffers from Fanconi's anemia. Hereditary non-polyposis colon cancer 200 out of 200, one person can suffer from deficient mismatch repair. Mismatch, that means some nucleotide is mismatched and it fails to repair. Warner syndrome, very, 1,000 out of 1,000 only 3% can suffer this type of problem. Deficient helicase, that means helicase enzyme is defective. And geroderma pigmentation, spot on the skin, 200 out of 250,000, 1% suffer from this. So deficient excision repair. So these are the diseases due to the disorders, but serious disease is cancer. Life exists, so DNA must be repairable. This is the real statement. How stable is DNA really? Thomas Lindahl started wondering towards the end of 1960s. That means Thomas Lindahl did the research in 1960s and discovered how stable DNA is, because at that time people believed that DNA is absolutely stable. At that time, scientific community believed that DNA molecule, the foundation of all life, was extremely resilient. Resilient means stable, not decay. Anything else was simply out of the question. But practically, Lindahl discovered that it is not out of the question. It is within the question, and evolution does require mutations, but only a limited number per generation. So in genetic information, if genetic information were too unstable, no multicellular organism would exist. This is the fact. During his postdoc at Princeton University, Lindahl worked with the RNA molecule, a molecular cousin to DNA. Why RNA molecule? Because it is single-stranded and it is easy to handle, and he used the bacteria to do that. And if it was well known that RNA was more sensitive than the DNA, because RNA is very sensitive and easily decay and destroy quickly if you offer the heat, and molecule can be break down. So he thought it is better to start with the cousin of DNA to get the real information. So DNA underwent a slow but noticeable decay. Thomas Lindahl quickly proved by using several experiments. And there must be molecular system for repairing all these DNA defects. He also made the conclusion by using his experiment. So he used the bacterial DNA and not the human DNA, because bacterial DNA is simple and small, and Lindahl and Sankar, they all use the bacterial DNA, and they found that yes, DNA damage can be taken place in the cell. And I can show you they discovered the base excision repair, which is very important for you to understand. Bit by bit, Lindahl pieced together a molecular image of, that means small by small effort, he confirmed that how base excision repair functions. Base excision repairs, that means if a defect at single base, how repair is taken place. Base excision repair also occurs in human beings, and Lindahl managed to recreate the human repair process in vitro. In vitro means laboratory condition, under control condition. In vivo means inside the human body. The decisive factor for Thomas Lindahl was the realization that DNA inevitably undergoes change even when the molecule is located in the cell's protective environment. And it had long been known that DNA can be damaged by environmental assaults such as UV radiation. This is why they did, some UV experiment. Here you can see the simple thing, mechanism. And this is the last slide. Please carefully follow how base excision taken place, and scientist Lindahl discovered it. Here you can see here guanine should bind with cytosine, but somehow wrong base uracil is wrong base is connected here. That means here there is a wrong base synthesis is taken place. That means it is a defect, single base defect taken place. In that case, DNA glycosylase present. This is an enzyme which can detect if there is any damage at the base level. It can detect where is the damage. This means one kind of proofreading. So DNA glycosylase, it identifies the damage and then removes base, leaving deoxyribose sugars, then removes it, AP site. You see, it is removed from here, thereby producing an abasic, that is purinic or pyrimidinic site by base flipping out. You see this flipping out, this part is cut, do surgery, endonuclease. This enzyme cut this part, this phosphate backbone. And then from the cell, the deoxyribose phosphate diesterase involved to cut this very nicely so that it can place a right base, that is cytosine, because guanine should bind with the cytosine and place cytosine here by DNA polymerase from the which is present in the cytoplasm or nucleus, and the ligase is used to paste it. So DNA polymerase replaces the missing nucleotide. DNA ligase seals this nick. Nick means this damage, and now it is repaired. So this is very important for your exam and for your understanding that single base, damage can be taken place in the DNA replication in the living cell, living organism, and there is a mechanism, several multi-steps mechanism to identify the damage, and then cut the double strand and clearly make the space for placing the right nucleotide to replace it, and ligase is used to paste it, very seal it, and make it completely correct DNA with appropriate sequence. No base error is here. So this is most important finding discovered by Thomas Lindell, and later I shall show you similar thing happened for several nucleotide wrongly mismatched nucleotide is even happened due to damage by radiation or other causes. Similarly, cut the wrong part by and also replace the nucleotide and seal the damage and repair the nucleotide damage in the DNA. So this is the base excision repair mechanism, very, very important for your exam also. And I hope you can easily identify, understand these steps how taken place. So I have given you the illustration, the simplest way, as well as the steps I described you. So if you are asked whether illustrate the base excision repair mechanism taken place in the cellular organism, then you can do it. This is the correct answer. Thank you very much for today, and I think it is better for with you to discuss a little more. And you can ask, it is your turn to ask some question, and if you do not ask the question, I shall ask you a question. So best idea is please ask some question so that you can get it clear.
Sir, আমাদের দেখে তো স্যার
Akbar Khan, right?
জি স্যার।
Okay.
স্যার বিলিয়ন সেল আছে স্যার, তো এগুলো যেহেতু রিপেয়ার মানে নষ্ট হলে আবার ডিএনএ গুলো রিপেয়ার হয় স্যার, তাহলে ঠিক কয়টা নষ্ট হলে স্যার বা কয়টা নিউক্লিয়াসের মধ্যে মানে কয়টা নষ্ট হলে স্যার একটা ক্যান্সার হওয়ার সম্ভাবনা থাকে?
This is a very, very critical question and very important question. Akbar Khan, I appreciate you for this nice question. Actually, the answer, is difficult because cancer can cause even a small, mutation, or a big mutation may not cause the cancer. It depends on where the mutation is taken place in your DNA sequence. If it is associated with a very important gene which is linked to the DNA replication, or a defect in the related to the apoptosis. Have you heard about apoptosis? Maybe not. In our cell, I told you, every 3 months our blood cell is replaced. That means where the old cell goes. Old cells are, killed, and there is a computerized like program. So it is called programmed cell death. There are several genes are involved in the programmed cell death. So all cells are died. But cancer cell, do you know that cancer cell never dies? In our body, in all organisms, cell has the mechanism to do apoptosis, that is programmed cell death. After certain period, cell needs to replace older cell by the new cell, and there are some genes involved there. But if apoptosis is as mutation taken place in the genes presence or genes involved in apoptosis, then there is high chance of cancer. And funny thing is that cancer cell never abide by the apoptosis, never dies. Only this is why they multiply and make a tumor-like structure. When they have no space in an organ after tumor, then they migrate to other organ. It is called metastasis. But naturally, all the cells in our organs, for example, your finger, there is a computerized program like system, how many cells should be there. But in case of there is a controlling mechanism. After that, no cell division will be taken place. Otherwise, your finger, if continuously grow the cell and multiply, your finger would be unusually big. But it never happens. There is a controlling mechanism. But in case of cancer cell, controlling mechanism of cell division is failed, and it is due to the mutation taken place in the programmed cell death or controlling mechanism of the cell. So a simple mutation may result the cancer if it taken place in such a sensitive point of the DNA. Thank you very much. It is a really a brilliant question. Anyone have any question? Yes.
Sir, can this DNA repair mechanism contradict with the gene editing technology that we use?
Interesting question. Yes, in case of gene editing, whether DNA repair mechanism is involved. Very, very brilliant question. Yes, when you cut somehow by radiation or by any ways, if you cut the double strand of DNA, as I mentioned, DNA is assaulted to damage. Immediately, DNA has the mechanism to repair. Immediately it has the repairing mechanism. And this repairing mechanism help us to do the editing. For example, if you target a base to defect a gene and you put the gene editing scissor, molecular scissor there to cut, and then immediately DNA has inherent mechanism to repair it, and it repaired, but that gene becomes defective, and gene expression would not taken place. If it is a bad gene, and if you somehow by using gene editing, you can make it defective, then it would not express. That means, gene editing taken place for the benefit of human. Yes, the principle of DNA repair mechanism we are largely been using for, gene editing. Thank you so much. Jerin Tasnim, good question. Very good question. Anyone have any question?
Sir, I have a question related mutation.
Yes, Abdullah Al
Moin Khan.
Moin Khan. Yes, please.
Sir, how to choose amino acid when inserting mutation, sir?
How do
Choose amino acid when inserting mutation mutation, sir. Inserting mutation.
if there is a mutation taken place in a particular gene, then it may be become defective in synthesis of mRNA. That is, its expression may be defected, or maybe wrongly synthesized the amino acid. It codes wrong amino acid to recruit in the protein. As a result, I showed you some physiological disorder taken place. Different diseases are taken place. Later, I shall show you some of the symptoms, and it causes physiological disorder in the cell, because wrong recruitment of the, if code is changed, and wrong recruitment of the amino acid can be taken place during the translation process, and it obviously hampers the cellular physiological process. And if it is a vital protein like enzyme involved in metabolic processes, then its consequences would be severe. But if it is non-protein amino acid or some other less important protein, then you can keep yourself safe. No cancer or no other big problem. Good question, Abdullah Al Moin Khan. And next, please ask some more interesting question.
Yes, sir. I have a question.
Mahjabin?
Yes, sir.
Firdaus, yes.
Yes, sir. Sir, normally DNA mutation occurs in some kind of cells, in cells. Like, sir, in a particular cell, we are
Yes.
having some DNA mutations. Then what's about the other cells that we are having in our body?
Very good, good question.
Sir,
Your
can't they fight those mutations?
yes, your question, if I understood well, DNA mutation, when we are exposed to any mutagenic agent or radiation, 100% cell are not damaged. Yes, only some cells DNA mutation taken place. How all the cells are affected? Actually, all the cells are not affected. They will work normally, but the cells which are affected, as I told you, cells are reproduced through programmed cell death. During cell division, when new DNA is synthesized, this defect create a serious problem, because it carries a wrong message, and then wrong DNA is synthesized if repair is not taken place properly, and then physiological disorder is taken place and cancer can be developed. But what happens if in few cells DNA damage taken place? 99.9% cases, cell has the repairing and maintenance mechanism to do in those cells, the affected cells. But other cells remain normal. They do not participate in this process. Is it correct? Majabin for those?
Yes, sir, I got this. Sir.
Okay, you got the answer. Thank you.
Thank you, sir.
Any more question?
Sir, I have a question.
Yes, Rojana Binte Azad.
Yes, sir.
Please.
So my question is, if the RNA is variable depending on time and environment, then genome can be variable depending on time and environment?
No. The answer is no, because RNA is a cousin of DNA. And when a particular gene is expressed, gene present in the DNA, then mRNA or RNA is synthesized. So it depends on the environment which gene will be expressed. But gene remains silent normally in the DNA. It is not always all the genes at a time never expressed. When it is necessary, for example, I am now talking, so some of the particular gene is now expressed. So a particular RNA is synthesized. But when I am not talking, I am not delivering the lecture, at that time, they are silent. But DNA, carries the hereditary information all the time, and they are, stable, mostly stable, but slightly unstable. I mentioned damage. If it is damage is stable somehow, then repairing, if it is not possible, sometimes bad consequences can be taken place or no consequences can be taken place. Is it clear now?
Yes, sir. So then, sir, when we do the genome sequencing, then the DNA information are only the main thing in that?
Yes, when we do the genome sequencing, as DNA is stable, so we can get the sequence of DNA. But if you do the RNA sequencing, for example, transcriptomic analysis, you every hour interval, if you do, you can get different. But DNA sequencing of human, for example, Homo sapiens, your DNA sequence and my DNA sequence of your friend's DNA sequence, 99.9999% same. This is why we are Homo sapiens, but slightly different because you and I, there are some differences in some characteristics. So in my lifetime, anytime if DNA sequence is taken place, it would be the almost same.
Thank you, sir. Got this question.
Oh, very good. Thank you. You are very brilliant. I am very much impressed because in my past experience since 2010, I didn't get such a brilliant question from my students. And this is, I am not sure. This is the, due to the environment is digital. In the classroom, I didn't find such a brilliant questions. Thank you very much. Any more question?
Sir, I had another question regarding
Yes, no problem.
gene editing. Sir, when gene is edited in a multicellular organism, when it is actually edited, I mean, is it only in the zygote phase, or can we do it in the further process when the organism is multicellular? I mean,
A very brilliant question. Yes, gene editing, we shall have a full-fledged lecture. I shall tell you the fascinating story of the gene editing, because yesterday, Dr. Tomalota Aditto, who died last night, she told me in the meeting that she mentioned me father of gene editing in Bangladesh, because we first, started work on gene editing in Bangladesh. Gene editing in the multicellular organism, your question is in which cell we do. In multicellular organism, we do in the target cell. Because lung cell never moves to the finger cell or like that. So if you want to, there is a defect in the certain gene which associated with the lung synthesis. So we do the DNA in the lung, DNA in the finger, everywhere, human DNA is same. So if you do the editing, then during DNA replication, the correction of particular gene would be happen, and all the cells will get the same DNA. And it is proven in the mouse as well as in many other multicellular animal model, even in the fish, even in the human. But human gene editing is strictly ethically prohibited till now, because gene editing is an emerging area. If any mistake we makes, then maybe our species would be in danger in this earth. This is why all scientists all over the world have a consensus that we should not do gene editing for human at this moment, although a Chinese scientist couple of years ago did gene editing in the human germline. Thank you, Jerin.
Thank you, sir.
Any more question? Brilliant.
Sir, how mutation is inherited into species if mutation occurs in a somatic cell chromosome?
How mutation is?
Inherited.
Good question. Mutation, always not inherited, but if mutation is stable, and if it is happened in the DNA, and if it is happened in a certain gene, then it is inherited. For example, many of the diseases are genetically inherited disease, even diabetes. Diabetes, genetically inherited, and several other diseases. So it can be inherited. And your question is, if it is happen in the somatic cell or reproductive cell, doesn't matter. Because if mutation is stable in the human DNA, and then, through the DNA replication process and change of the cell, it moves to the all DNA, all the cells. And then it also moves to the reproductive cells. So the cell has some homeostasis mechanism. So it shares same DNA sequence in all the cells. But it is very tough for you to understand. Even a single cell from your finger and maybe from your from your hand, maybe if you single cell DNA, if you do, you may find some differences. Such differences are unusually small and negligible, no clear phenotype we can see. And always, balancing and, all the DNA, how they can maintain in all the cells, somatic or a reproductive cell, same sequence, there is some also mechanism. Otherwise, there would be a big chaos.
Yes, sir, I understood that.
Thank you very much. Big chaos, for example, if DNA in my finger, DNA in my elbow, or DNA in my head different, they express differently and create a serious problem. For example, if a country, Bangladesh, has 5 prime ministers, what would happen? We have Sheikh Hasina is the prime minister, and she can do undo anything, whatever she likes, and she is the chief executive. But if 4 Sheikh Hasina in our country in different region, what would happen? You can imagine the chaos you cannot mitigate. And similarly, in the cellular system, there is some governing system. I told you, program cell death, program gene expression, everything is programmed and very nicely. For example, a person cannot, certain age, for example, you cannot be taller from now after 2 years or 3 years. At certain age, you will grow, but after that, you will not grow and you will stop. This is, some program in the DNA, and it is the secret. And all of the mechanisms we are not yet discovered. Any question?
Sir, I have one. If it's
A brilliant
silly
question.
Sir, can I ask a silly question?
And today attendance also very good. I am very happy. And if you have no question.
Assalamu alaikum, sir.
Yes, Tanvir.
স্যার আমার একটা কোশ্চেন ছিল স্যার, যে আমরা নিউজে আমি দেখছিলাম স্যার, যে করোনা ভাইরাস স্যার অনেক রূপ চেঞ্জ করে স্যার। এই যে যেমন বাংলাদেশে যে করোনা ভাইরাসটা আসছে, সেটা লেস সিভিয়ার দেন যেটা আমাদের যেটা ইউএসএ তে অ্যাটাক করছে কিংবা ইউরোপে অ্যাটাক করছে, তা এটা কি কোনো ধরনের মিউটেশন যে চেঞ্জ হয়ে যাওয়া করোনা ভাইরাসের টাইপটা?
It is a very good question. Very good question. In case of coronavirus is a single-stranded, RNA virus,
RNA
and
virus.
it is coated by several, spike proteins and envelope protein, nucleoprotein, and it has the mechanism to bind with SCE receptor in a human, cell, and in our, respiratory tract. And it can enter into the body, and coronavirus itself cannot replicate its RNA. So what it does, it uses human cell and transcription and translation machinery to replicate it, to make its copy. And during copying mechanism, this coronavirus has some inefficiency of proofreading exactly whether the parent one and the daughters, all the huge copies they make, and this is why always mutation is taken place. This is why there are more than 100,000 of coronavirus sequence are now deposited in the global gene bank, and there are lots of variation. From our Bangladesh, 200 RNA sequence of coronavirus have been deposited in the global gene bank, and it is found that the coronavirus in Bangladesh, coronavirus in other countries, there are some differences, and it is not yet clear such differences are involved in their virulence or severity to the human, because this is a new coronavirus. 80% of the sequence are known, more than 20% sequence are unknown. So scientists are dissecting and understanding the detailed mechanism, and it takes a little more time to understand which sequence are crucial for its virulence. Previously there were two coronaviruses. One is SARS, another one is MERS, and those two coronavirus was severe, and death case was very high. But what happened after 2 years, when vaccine was nearly developed, they were disappeared. They lost their virulence capacity, that is capacity to infect the human. Even in case of SARS-CoV-2, it may happen, but its biology is still poorly understood. Thank you very much. Very nice question.
স্যার আরেকটা ব্যাপার স্যার, এই যে স্যার করোনা ভাইরাস কন্টিনিউয়াসলি মিউটেশন হচ্ছে, কন্টিনিউয়াসলি তাদের মানে চেঞ্জ হচ্ছে, ফরমেশন চেঞ্জ হচ্ছে, তো আমরা স্যার রিসেন্টলি অনেকগুলো ভাইরাস তৈরি করতেছে, তো স্যার এমন মিউটেশন পর্যায়ে এমন হতে পারে যে এমন একটা করোনা ভাইরাস তৈরি হলো, একটা আলাদা একটা টাইপ, যেটাই ভাইরাস এই যে ভ্যাকসিনটা তৈরি হচ্ছে, এই ভ্যাকসিন সেটাকে প্রতিরোধ করতে পারতেছে না, কিংবা যেহেতু অনেকগুলো টাইপের হচ্ছে, তো স্যার এটা কিভাবে
It's a valid question. When I was in USA as a Fulbright visiting professor in 2017, immediately they asked me to take a flu vaccine, flu shot. They called flu shot. That means vaccine against influenza or flu. And I asked them how much is it effective for my whole life. They said no. Its efficiency is 60% or 50%, because influenza virus, they can mutate very quickly. So in case of coronavirus, the vaccines which are being developed or under the process of development, there is no guarantee that it can protect us. The vaccine is designed in a way so that a big coverage of mutation could be tackled. But even though if new strain is developed which is not covered by this by the vaccine, developed vaccine, then it may be problematic, and vaccine may be ineffective, and it is possible in case of coronavirus. But we are hopeful that vaccine production is moving very fastly, and we are no way to hope to get the vaccine in future, maybe next year.
থ্যাংক ইউ স্যার। স্যার আরেকটা ব্যাপার হলো কি স্যার, স্যার আমরা কি একটা একটা কোম্পানির একটা শট নিয়ে মানে নিয়ে কি করোনা ভাইরাসকে প্রতিরোধ করতে পারবো, কিংবা আমাদের রাশিয়ার করোনা ভাইরাস নিতে ভ্যাকসিন নিতে হচ্ছে, চায়নার ভ্যাকসিন নিতে হচ্ছে, আমেরিকার ভ্যাকসিন নিতে হচ্ছে, ইন্ডিয়ার ভ্যাকসিন, দেখা যাচ্ছে সবগুলো নিয়ে তারপরে আমরা ফুললি প্রতিরোধ করতে পারছি কিংবা বিঘার বিঘার কভারেজ পাচ্ছি, এরকম তো টেন্ডেন্সি দেখা যাবে স্যার যে হ্যাঁ, আমি একটু বেশি রিস্ক ফ্রি থাকি, এই জন্য আমি 2-3টা ভ্যাকসিন নিয়ে নিলাম।
No,
এটা কি
no,
প্রয়োজন থাকবে?
it is meaningless. If a vaccine is, effective, then, it will induce enough defense system, antibodies, and other T cells against the coronavirus. So no need to take 4 vaccine together, because, for example, if you then it would be problematic. Your body cannot tackle it, because even for sleeping pill, if you take too many sleeping pill, you maybe sleep forever. So in case of vaccine also, I think there would be some doses, how many times you need to take the vaccine, maybe 2 or 3 shots or like that. Same vaccine would be the wise. But if it is proved that China vaccine is not no more effective, maybe second vaccine we may choose. But availability of the vaccine for 7.3 billion population in the earth would be really a big challenge, which we are not yet considered. So availability of vaccine to all the population of our country in any vaccine, it would be a big challenge. But let us hope for the best.
Thank you, sir.
Thank you very much. Any more question? If no more question, I would like to stop here, and I wish all of you, safe and sound health to fight against the ongoing pandemic coronavirus disease. And please enjoy learning the biotechnology. Whenever you have a question, please don't hesitate to ask me, and I am very happy with you, because you are very interactive students. Thank you so much.